Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Sophia Krombholz. Listeners, Sophia is a PCC Fellow and a research scientist and research associate at the Manfred Donike Institute for Doping Analysis in Cologne, Germany. Today, we're excited to discuss Sophia's career path, her PCC-funded research project, and other exciting anti-doping research. So welcome to the show today, Sophia. How are you?
I'm good. Very good. Thanks for having me.
Well, we are thrilled to have you on the show, and we're looking forward to learning more about you and the work that you do. So, Sophia, can you perhaps start by telling us a little bit more about yourself?
Sure. So, as you already said, I'm currently working as a scientist at the Manfred Donike Institute for Doping Analysis in Cologne. And I'm there as a postdoctoral researcher and also fellow of the PCC. And within our research department, I primarily work with liquid chromatography, mass spectrometry, investigating the metabolism and excretion profiles of doping substances, also supervise postgraduates. And yeah, that's me.
Fantastic. And I know when I talk to people in the field, I'm always curious to learn more about how they ended up where they are. So, Sophia, can you tell us what first got you interested in anti-doping? And perhaps could you share the path that you took to get to your current positions?
So I studied pharmacy at the University of Bonn. And I guess I got interested in analytical chemistry quite early on, more particularly also forensic toxicology. So kind of the possibility of just detecting the smallest amount of a substance in biological samples and also the techniques behind it. And then I think it was actually an article in the Journal of the German Pharmaceutical Society in which I read about the work of the anti-doping lab in Cologne. And I thought, well, that's also really exciting. So after university, I did this so-called practical year, which is something you need to do in Germany to get your official license as a pharmacist. And I spent a part of that in a pharmaceutical company doing some stability studies. So I kind of gathered some more experience with analytics. And that encouraged me to actually pursue a career in analytical chemistry. During that time, I got into contact with Professor Mario Thieves at the German Sport University in Cologne about a PhD position. And luckily that worked out. And I started doing research here in 2020 and was very fortunate actually to work on quite a lot of different projects during my time as PhD student and didn't really hesitate much as the opportunity came up to continue my research here as a postdoc.
So maybe could you tell us a little bit more about what was your experience like completing your PhD at the German Sport University of Cologne, a prestigious institution, with your mentor, Dr. Mario Thieves, who I think everybody in this field knows?
It's been really a great experience. I guess we have quite a big research department and also different topics that we're dealing with. So I think I got to learn from really great and experienced researchers there.
Definitely. And Sophia, perhaps next, could you tell us a little bit more about the Manfred Donike Institute for Doping Analysis, where you're working right now?
Sure. The Manfred Donike Institute for Doping Analysis, or MDI short, I think has officially been founded in 1996 to kind of continue the life work of Professor Dr. Manfred Donike, who really was a pioneer of doping analysis. And the MDI is affiliated to the Institute of Biochemistry of the German Sport University in Cologne. And this is actually one of the oldest doping control laboratories in the world. So on the one side, we focus on the analysis of doping control samples, of course. We analyze more than 30,000 human and also over 4,000 equine samples per year. But as I just mentioned, we also have a big research department. So kind of work a lot on the improvement of detection methods, the development of new detection methods, lots of different topics.
Very cool. And of course, we mentioned that you are a PCC research fellow. So, Sophia, can you tell us, how did you originally find out about the Partnership for Clean Competition, this PCC fellowship, and then ultimately decide to pursue the fellowship?
Well, in my case, it was actually my mentor, Mario Thieves, who told me about the fellowship and who also encouraged me to apply. So at that time, we already had a specific project in mind that we wanted to pursue. And fortunately, the PCC liked the idea too. And I actually thought it was great because this kind of offers me the chance to work on my own research project for at least 2 years now and gain some more experience.
Absolutely. And what was the application process like for you?
I think it was pretty similar than applying for a grant funding. So I could kind of adapt to that in a way. And of course, I had support from Mario. So I think the first step was a pre-application, which included, of course, my biography as well as Mario's, and also him supporting me, and then like a short description of what we have planned. Then we got feedback from the PCC, and then we could send in a full application, which, as I mentioned, is kind of like the grant application. So includes also a timeline of the major milestones, what exactly we have planned, etc.
Very good. It sounds relatively painless, and I'm so glad you were selected for this PCC fellowship. And perhaps next we could talk a little bit more about your PCC fellowship project. I know your PCC-funded fellowship project has been investigating the metabolism of anabolic steroids taken through alternative routes. So not through the typical oral tablets that people may think of or intramuscular injections. So Sophia, can you tell us more about these alternative administration methods that you've been looking at?
Yeah, of course. So one route of administration that we are particularly interested in is transdermal administration. So meaning absorption of doping substances via the skin. And while this is not a new concept for anti-doping, so we know that, for example, transdermal testosterone has been available since the 1980s, this has somehow become relevant from another perspective recently. So there have been several cases where athletes have attributed the presence of prohibited substances in their sample to unintentional and also unknowing contamination via the skin. So we know that generally in doping control testing, we have quite sensitive analytical methods and, of course, cover a broad spectrum of chemicals. And also athletes are tested quite frequently. So this can all increase the risk of adverse analytical findings that are caused by contamination other than deliberate doping. And so in that regard, skin can be another potential route of exposure. So if we want to be able to evaluate the plausibility of such claims, we of course need data on skin absorption, but also, if possible, on the biotransformation and the excretion of doping substances that were administered via the skin. So yeah, that is one major reason why we are interested in that. But on the other hand, we also have to consider that the transdermal route is deliberately used by athletes to administer performance-enhancing drugs. So probably for all the reasons therapeutics are administered that way. So, for example, because it's more convenient than an injection or there are fewer side effects. And since there are some fundamental differences pharmacokinetically between, well, let's say oral ingestion of a steroid or the transdermal absorption, we kind of need to investigate just the influence on the excretion profile, but also the metabolites that we usually aim to detect in doping control urine samples. And yeah, I guess another thing is that there were claims that within the systematic doping in Russia related to the Sochi Olympic Games, transdermal and also buccal steroid administration, so the absorption via the oral mucosa, were deliberately used to avoid the formation of certain metabolites and in that way to undermine doping control testing. So apart from the transdermal route, we're also looking into buccal steroid ingestion and the influence on the excretion and the metabolite profile in urine.
That makes sense. And we talked a little bit about the justification of why these alternative routes of administration are important to investigate. And I think it's really interesting to think about how these drugs or prohibited substances might be entering the body. But taking a step back, why might you suspect that the route of administration could impact whether you can detect use of a particular substance or not when you're looking at an anti-doping sample at the end of the day?
For example, if you administer a steroid orally, you usually have a huge impact of the so-called first-pass effect. That means that a large portion of the dose is actually metabolized before even entering the systemic circulation. That is something that is actually avoided by transdermal or buccal administration. So that's just one reason why this might have an influence on the metabolites or the amount of metabolites that we can detect in the urine samples. And in addition to that, there are also differences in the kinetics of the absorption. So usually transdermal absorption is much slower. Buccal absorption can be quite fast. And also the substances are usually eliminated more quickly. So this is why this could have definitely an influence on the detection windows.
That makes sense. So then let's talk about where you are in this project to date. Sophia, do you have updates you can share with us on how things are going with the project so far?
Yeah, sure. So in this particular project, we were doing a study using the anabolic steroid trembolone, kind of as a model substance. And we did an administration study with male volunteers who then either applied the drug orally in a capsule or transdermally in a cream on the lower forearm or buccally. So we had the volunteers swish around an alcoholic solution of the steroid in their mouth just for a couple of seconds. And then they collected urine samples, which we could analyze by LC-MS. We know that usually trembolone is metabolized quite extensively after oral ingestion. And it was quite interesting to see that all the metabolites that we were looking for were also detected after transdermal and buccal administration. So eventually those seemed to be detectable kind of independent of the way they were or trembolone was ingested. But of course, the intensities and also the excretion profiles we obtained were quite different between the study groups. So kind of expectedly, the absorption via the skin was much slower. The peak concentrations were also lower, but we could actually see a prolonged elimination. On the other hand, after just these few seconds of buccal steroid absorption, the peak concentrations were actually higher than after transdermal administration. But on the other hand, the detection windows then were much shorter. So this is something that we can probably also transfer to other steroids. When we had a look on the differences, especially in the metabolism, there was actually one metabolite that showed a relatively higher formation after transdermal and buccal administration. And this is something that can become interesting or more relevant, particularly if you have a case of suspected or claimed contamination via the skin. And then you have to assess whether this is plausible or not. And yeah, I guess that's the current state, and something I'm currently working on is trying to synthesize this metabolite and to find out more about its structure.
Very cool. Now, to back up a little bit, Sophia, can you explain why trembolone specifically was selected as the steroid that you would look at first? I know you mentioned this route of administration question is relevant for a variety of different steroids.
So for once, it's a steroid that is still quite frequently used. And it was also one of the steroids that was contained in this cocktail that was described related to the Sochi doping allegations. And there are some other structurally similar steroids, such as nandrolone. So we thought this might be actually a good model substance here.
Excellent. And you mentioned that the steroid trembolone was then administered transdermally, buccally, and then also orally in a capsule kind of as a control. And then you looked at the metabolism of the steroid after it was administered using these different methods. Did you use the same dose across all these three different administration methods, or how did you decide what dose to use in terms of what would make sense for each of them?
That's an interesting question, actually. So we decided to do the same dose for all three routes of administration, just to have comparable data. But of course, it would be interesting to see the actual differences in bioavailability. But for that, we would need blood data to estimate that.
Absolutely. And Sophia, I know you hinted that at this moment you are trying to synthesize this metabolite of interest. And I know there are always challenges that you run into in science. So have you encountered particular challenges, or perhaps could you describe some of the things that you've learned along the way in this project?
Yeah, I guess research itself can definitely be challenging. Usually things don't work out the way you expect them to, or I don't know, whenever you have time pressure, there's some instrument dropping out and leaving you alone. I guess what I learned is that there's always someone in the lab with maybe more experience who can eventually help you.
I think that is an important lesson, just when to ask for help. So it's great to hear more about how the project is going so far. And let's talk about the potential impacts of this project and some of the results that you're seeing so far. So how do you see this line of research or this particular project being relevant or potentially being applied in the field of anti-doping, Sophia?
I guess eventually this data is, of course, more directly available for trenbolone-related AAFs, but I guess more importantly, it kind of provides the basis for the general evaluation of cases where there is suspicion of transdermal steroid application. As I said, for example, also with steroids that show structural similarity, for example, nandrolone. So this kind of research can really help in the result management process to better evaluate and assess these cases of alleged or possible contamination.
Excellent. And you mentioned that the current status is looking for and trying to synthesize a particular metabolite of interest. So I guess looking forward, what are some of the next steps of things that you'd like to work on once that is completed?
Something that we're also considering is doing microdosing experiments to see if that also leads to detectable amounts of substance, and if there are maybe also ways to differentiate contaminations from deliberate doping. So generally just maybe use the analytical data to better estimate the dose and time point of intake. I guess another field of research that we're looking into potential in vitro models to simulate transdermal drug absorption, because these kind of models can really be useful, particularly for new substances where we have no data yet or that often also lack approval for human use.
Absolutely. And Sophia, it sounds like you've made some really great progress in this project so far. So can you tell us a little bit more about what this overall fellowship experience has been like for you so far?
Yeah, I think for me, it's been really a great experience. Not only the project I just talked about, but also just generally working here at the Institute and still learning new things and developing my skills. And of course, also the recent conference in New York City was definitely a highlight for me, where I could present my own data, but also get to know just many faces from the PCC and from the global anti-doping work, and of course, listening to exciting presentations.
Absolutely. And your conference presentation was a hit at the event, Sophia. So let's talk about that next. You presented in one of the sessions at the recent PCC conference this year. So what was it like for you being on stage and being able to share your research with the diverse expert audience that was there in attendance?
Oh, it's been really nice presenting there. I really hope the audience enjoyed it too. But yeah, it was especially also interesting to not only listen and talk about the laboratory aspect of anti-doping work, but also to hear experience from representatives of the major leagues, the IGA, USADA, and so on. And yeah, I think I especially loved hearing from Shannon Robery, who talked about her experience as an athlete and being awarded the bronze medal for the London Olympics after so much time. That was actually really motivating, I think, and somehow gives you just a bigger picture of the global anti-doping work and kind of reminds you what you are doing the research for eventually.
Definitely. I think it highlights and brings to the forefront the importance of the work that you are doing in the lab every day and just the value that it has for athletes. So is there another research project that you are working on at the moment, Sophia, that you are really excited about and want to share with all of us today?
Yeah, something I'm also enjoying a lot is a project we're doing on some smaller peptides that have been introduced to the prohibited list in 2024. And we're kind of looking into stability of these peptides and potential degradation. Of course, this is quite different to looking at small molecules or steroid metabolites, but kind of like doing that too.
And could you share some details about maybe how it's different when you're thinking about how you have to analyze or work with these small peptides versus how you're analyzing or working with steroids?
I guess they're just generally very challenging when it comes to stability, which is just something you always have to keep in mind while preparing the samples, like not to use any glass equipment and stuff like that. And just the way they are degraded or what you're looking for is quite different and can be actually quite complex if you're looking at post-translation modifications and stuff like that. I guess you also need a lot more software tools to do that. So yeah, but it's interesting.
And then thinking about the big picture for this small peptide analysis project, can you share with us what is the big question that you're trying to answer? What are some of the problems that you're trying to solve in the work that you're doing in this area?
So it's been suggested in previous studies that these are also degrading quite fast if administered to humans or to animals. So we're kind of hoping if we have a closer look at the degradation, that we can maybe find some degradation products or metabolites that we can try to detect in our doping control screening, and thereby also just kind of increase the chance of detecting a potential misuse of these peptides.
That makes sense. And then to give our listeners some context, could you share some examples of what are these smaller peptides that you're looking at?
We are currently looking at these are called kisspeptins. So they are also involved in the gonadotropin releasing hormone, leading to increased concentration of testosterone. That's why they are prohibited. And those are also endogenous peptides, but there are also synthetic versions currently under research, and they are supposed to be used in, for example, hormone-related cancers.
That makes sense. So they have this legitimate medical use, these small proteins, but people are starting to use them in the world of sport for doping as well. So, Sophia, we know from earlier when we talked about your path to a career in anti-doping that you have a background in pharmacy. And I think this is really cool. And I think people coming into anti-doping, bringing a variety of different backgrounds and different expertise brings a lot of value to the field. So can you share your thoughts on just the interdisciplinary nature of anti-doping research and how your background specifically in pharmacy maybe intersects with other disciplines in the field?
Yeah, that's actually something I really like about anti-doping research, because, of course, a lot of the research that we are doing has a lot to do with analytical chemistry. So I guess a lot of the people, at least in our lab, have a background in chemistry or food chemistry. But over the last decades, the range of substances and methods that we're looking into has become so much wider, including peptides, proteins, but also complex methods like blood doping or gene doping. And therefore, you also need scientists with experience in biology or microbiology. And studying pharmacy in Germany involves a lot of chemistry, but you also kind of acquire a fundamental knowledge of drugs in general and their pharmacokinetics and biotransformation, which I guess are also important aspects of anti-doping research. For example, looking at my postdoc research project, I had to deal a lot with the structure and biology of the human skin and factors influencing skin absorption and so on. And essentially, those were all things I had been dealing with previously in university. So it was really helpful. I guess you could say just generally that you really need expertise from all possible disciplines to be able to detect the entire spectrum of analytes in the best possible way.
Well, Sophia, I think it is wonderful that you found your way to this field of anti-doping. And I know in science, in this early career stage, it's very common to face a variety of different challenges in this early career stage, regardless of what field you end up in. But Sophia, could you share with us your thoughts on what are some of the biggest challenges that you've faced as an early career anti-doping researcher?
Honestly, so far, I've been really lucky because I always had a lot of support from great and experienced scientists around me. So this has been really helpful. And I guess that's something everyone can take home, that I guess great colleagues and people around you can really help you get where you want to be.
Certainly. And I think for you specifically, you mentioned earlier that you began your PhD journey in 2020, which was a tumultuous year for, I think, everybody. So can you share with us, how did the global pandemic affect your research?
Yeah, I guess it was kind of an interesting and difficult time for everyone. We were kind of lucky because most of the time we could actually come to the lab, but of course it was really different. We had to wear a mask and couldn't do lunch breaks together or something like that. And it was different, but fortunately it was over at some point, and then I could get to know all the colleagues a little bit better. And that was definitely more fun.
Yeah, I think that moment where you finally see the other half of everybody's face is a revelation.
Yeah.
Well, Sophia, you are doing remarkable work in the field. And for our listeners out there who may be considering applying for a PCC fellowship or just getting involved in anti-doping research, can you share your thoughts on what are some of the benefits?
Yeah, I guess for me, the conference was already a huge benefit. But besides that, it's just, I think, a really good program that finances postdoc scientists in anti-doping research. That also includes the opportunity to work in a WADA accredited lab and also to gain practical knowledge. Because I guess anti-doping research is a bit of a niche between, bioanalytics and sports scientists. And I think it's really great that there's a program that supports and funds exactly that. If you're considering a career in anti-doping or researching in the field of anti-doping as a postdoc scientist, I could really recommend the program.
Absolutely. And have you had any contact with or have you had a chance to meet any of these other PCC fellows, whether they were current or previous PCC fellows?
Yeah, I actually had the chance to meet both Sarah and Jenna here in Cologne at the Manfred Donike workshop, but also again in New York. And yeah, it's always nice to get into contact with others kind of around the world.
Definitely. And Sophia, I know there are many challenges and problems that remain in the field of anti-doping, and the field is just evolving rapidly. So if we gave you all of the resources you could possibly dream up, is there one particular problem in anti-doping that you would most want to solve, or a research question that you would want to address first?
Yeah, well, I know this is a complex issue, but since I've been working on LC-MS-based intact protein analysis during my PhD, I guess it has always been kind of intriguing to work on an LC-MS-based analysis of EPO, or at least kind of look into that a little further.
I like it. And Sophia, why is this question of particular interest for you?
For once, it's really kind of cool to be able to measure intact proteins by LC-MS, because this is, I guess, still kind of new. Usually large proteins are measured based on immunoaffinity methods. And EPO, I guess everyone knows that, is a challenging protein, particularly challenging to differentiate the recombinant and endogenous EPO already with the method that is currently available. And I think it would just be really cool to manage that by LC-MS.
Interesting. So I'm curious, Sophia, if you decided you wanted to do an analysis of EPO using LC-MS today, what are the barriers? What are the things that are stopping you or preventing you from doing it?
Well, I haven't done it so far. No, as far as I know, it's a glycoprotein that can be already pretty difficult looking at the chromatography to separate that and to analyze it. Then again, you also need the sensitivity to actually measure it in blood samples eventually. So yeah, I guess you need kind of advanced techniques in chromatography. You need the analytical sensitivity. As I said, it's a complex issue.
Well, a dream project to look forward to perhaps someday, Sophia. And Sophia, we appreciate you sharing your experiences and talking more about your PCC Fellowship research project and other research today. If our listeners are curious and they want to learn more about you and your work, what is the best way for them to do so?
Well, I'm on LinkedIn, of course. You can find me there, but also find updates and all my publications on ResearchGate too. So feel free to check that out.
Perfect. Well, listeners, definitely take a moment to get connected with Sophia on LinkedIn or on ResearchGate and learn more about the amazing work that she's doing. And Sophia, it's been such a pleasure to chat with you on the show today. Thank you so much for your time.
Thank you so much. It's been nice.
Wonderful. And listeners, we always appreciate having you here with us as well. If you haven't had a chance yet, please take a moment to subscribe to our podcast and leave a rating or review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.